Good morning, dear students. Welcome to our zoology online class. Hope you are very happy. Okay, dear students. In the last class, we started the third chapter. Our lesson's name is reproductive health. And in the last class, we studied about one very important 5-mark question: birth control. Different types of birth control methods. So in that last video, I clearly explained different types of birth control: permanent birth control, then the barrier birth control, then the natural methods of birth control. Like that, different types of birth control methods are there. So we have studied all these types in the last class. Okay, the students, today we are going to study about two more topics. Okay, from third chapter, today we are going to study about two topics. Okay, so the first topic is assisted reproductive technology, and the second topic is detection of fetal disorder during early pregnancy. So today we are going to study about two important topics. The first topic is ART. ART meaning assisted reproductive technology. Okay, assisted reproductive technology. Okay, dear students, listen very carefully. So this is 5-mark questions and many 2-mark and 3-mark questions and 1-mark questions also there. So listen very carefully. So assisted reproductive technology meaning many procedures. Okay, procedures which are used to handle the gamete or embryo outside the body to achieve a successful pregnancy is called assisted reproductive technology. Okay, so many procedures are involved in this technique. So a collection of procedures which are used to handle the gamete or embryo outside the body to achieve a successful pregnancy is called assisted reproductive technology. In short, we can say ART. ART. Okay, there are a lot of techniques are there. So now we are going to study about all these techniques one by one. Listen very carefully. The first technique is intrauterine insemination, IUI. In short, we can say IUI, intrauterine insemination. Here, intrauterine insemination meaning maximum this technique is used to treat the man with less sperm count. Okay, so in some person's body, the amount of sperm is or the production of sperm is very low. Okay, so for that people, this is the very best technique. So in this technique, the sperm is collected from the husband or from a donor, another person donor. Okay, then the sperm is directly inserted inside the female reproductive system. Okay, inside the uterus of the female reproductive system. Understand of you? So when the sperm is inserted or introduced inside the female reproductive system or in the uterus, automatically it is fused with the egg or ovum of the particular woman. Understand? Then after that, the zygote is forming, the zygote is slightly coming and attached with the uterus. Then finally normal pregnancy takes place. So here intrauterine insemination meaning this technique is used to treat the person with low or less sperm count. Okay, or infertility person. Here in this technique, the sperm is collected from the husband or from a healthy donor. Then the sperm is inserted inside the uterus region of the female reproductive system. At the same time, the ovary also stimulated to produce the ovum. Both ovum and egg are joined in the fallopian tube. The sperm which are inserted inside the uterus is moving to the fallopian tube. Okay, then the ovum also coming from the ovary to the fallopian tube. So in the fallopian tube, both sperm and ovum both are joining and the fertilization process takes place. Understand of you? So this technique is called intrauterine insemination. The next technique is called in vitro fertilization. In short, we can say test tube baby. Okay, in vitro fertilization meaning test tube baby, commonly called test tube baby. Okay, so in this technique also both the sperm and ovum or egg are allowed to fuse in the outside body. Okay, that means in a test tube. The sperm and eggs are allowed to or fused or joined togetherly in a test tube. So that only it is called test tube baby. Actually, this in vitro fertilization techniques was once upon a time used to treat the women with some problematic fallopian tube or damaged fallopian tube or infection fallopian tube. Okay, for this purpose only this in vitro fertilization techniques was used once upon a time. But nowadays this in vitro fertilization is used to treat maximum infertility cases. Okay, for maximum infertility cases, maximum people using this in vitro fertilization technique. That means test tube baby technique. Understand this, friends? So this is a small introduction about this in vitro fertilization technique. Then there are a lot of some steps are involved in this in vitro fertilization. So listen very carefully. So there are 5 steps are involved in this in vitro fertilization. First one is ovarian stimulation. Second one is egg retrieval, fertilization, embryo culture, and embryo transfer. So these are the 5 steps involved in the in vitro fertilization. So this is one of the very important 3-mark questions. So listen very carefully. So here ovarian stimulation meaning. First, human chorionic gonadotrophic hormone is injected to the person, to the mother or woman. Understand you? So when this human chorionic gonadotrophic hormone is injected to her body, automatically the ovary started to release the ovum. Okay, ovary started to release the ovum. Then after 34 to 37 hours, the egg or ovum is collected from the ovary by using a small surgery, minor surgery. Understand you? So the first step, human chorionic gonadotrophic hormone is injected to the woman's body. Then next in the second step, after 34 to 37 hours, the egg or ovum is collected from the ovary by doing a minor surgery. For this surgery also, before doing surgery, anesthesia should be given. So this is second step. This step is called egg retrieval. Okay. Then the outer surface of the egg, some small unwanted tissues are there. Every tissues or unwanted tissues are removed from the outer surface of the egg. Then the third step, fertilization. Third step is the fertilization. Now this egg is kept along with the sperm in a medium, in a culture medium. Understand? Here one important point you have to remember. Maximum for each and every egg, 10,000 to 1 lakh sperms are needed. So in a culture medium, if we keep one egg to fertilize this single egg, maximum 10,000 to 1 lakh sperms we have to add in the medium. Then only any one sperm can enter inside the egg and the egg is converted into fertilized egg. So now we completed the third step, fertilization, meaning the collected egg is kept along with the sperm. Here one point I said to you, here maximum 10,000 to 1 lakh egg is added, or we have to keep along with the single egg for effective fertilization. The next step is embryo culture. Now both sperms and the eggs are fused together and now the egg is called fertilized egg. This fertilized egg is allowed to divide into 8 cell stage, 8 plastomere stage. So this technique is called embryo culture. And the last technique is embryo transfer. Okay. Now this 8 cell stage egg or 8 plastomere stage eggs are now transferred to the uterus of the mother. When this egg is transferred to the uterus of the mother, automatically it will attach to the wall of the uterus and it is developed into a baby or fetus. So these are the 5 steps involved in the in vitro fertilization. So in vitro fertilization meaning the fusion of both egg and sperm outside the body in a test tube or in a lab is called in vitro fertilization. And once upon a time this technique is used to treat the female or woman with defective fallopian tube or damaged fallopian tube or the infected fallopian tube. But nowadays for maximum infertility cases, this in vitro fertilization is the one and only solution. Okay, and there are 5 steps are involved in this in vitro fertilization. First one is the ovarian stimulation. Here, human chorionic gonadotropin hormone is injected to the female. The next step, egg retrieval. After 34 to 37 hours, the ovary is stimulated. Then now the ovary is ready to release the egg. Now the egg is collected from the ovary. Then the unwanted tissues are removed from the ovary. Egg. Then third step, fertilization. The collected egg is kept along with the sperm. Maximum 10,000 to 1 lakhs of sperms you have to add in the medium. Then fertilization. So if here the fertilization process, both eggs and sperms are joined together and zygote is formed. The next step, embryo culture. Now the zygote is allowed to divide into 8 plastomere stages. Okay, and the last step, embryo transfer. Now this 8-celled plastomere stage zygote is transferred to the uterus of the mother to develop into a well-developed fetus. Understand, my dear students? So these are some of the steps involved in the in vitro fertilization. So this is very important for pre-mark. Then next, so now we have completed two assisted reproductive techniques. The first one is intrauterine insemination. Second one is in vitro fertilization. Now third, we are going to study about zygote intrafallopian transfer. In short, ZIFT. Okay, zygote intrafallopian transfer. So in this technique, the zygote, which is in 8-cell stage, okay, 8-celled plastomere stage, zygote is directly inserted inside the fallopian tube of the female reproductive system. Listen very carefully. So the 8-celled plastomere stage zygote is inserted inside the fallopian tube of the female reproductive system. From the fallopian tube, it is slowly moving and finally attached to the wall of the uterus to develop a fetus. Understand? So this is the meaning of zygote intra-transfer fallopian transfer technique. So three assisted reproductive techniques we have completed now. Then fourth one is intrauterine transfer, IUT, intrauterine transfer. Here in this technique, the zygote with more than 8 plastomere stage, okay, more than 8 plastomere stage zygote is transferred to the uterus region of the female reproductive system to develop into a fetus. Listen up you. So I hope you all understood the difference between these two techniques. So in zygote intrafallopian transfer, 8-celled plastomere stage zygote is transferred to the fallopian tube. But in intrauterine transfer, the zygote with more than 8-celled plastomere stage zygote is transferred to the uterus of the female reproductive system. Understand, my dear students? Then next one is gamete. Next technique is gamete intrafallopian transfer. See, not zygote, gamete. Okay, gamete intrafallopian transfer. Here, gamete intrafallopian transfer meaning in this method, the egg is collected from the donor mother and the sperm is collected from the donor father. So, egg is collected from the female and the sperm is collected from the male. So, both egg and sperms are simultaneously placed in any one of the fallopian tube for the fertilization process. Okay, so egg is collected from one person, sperm is collected from another one person. So, both egg and sperms are simultaneously kept in any one of the fallopian tube of the female reproductive system. So inside the female reproductive system, both the egg and cat are fusing together and zygote is forming. The zygote is moving to the uterus and it is attached to the uterus wall to develop into a fetus. So this is the meaning of this gamete intrafallopian transfer. Okay, gamete intrafallopian transfer. Then next technique is intracytoplasmic sperm injection. Intracytoplasmic sperm injection. Listen very carefully. In this technique, only one sperm is injected inside the cytoplasm of the egg. So just imagine it's a egg. So in this region, cytoplasm region is there. The center of the nucleus is there. So one sperm is directly inserted inside the cytoplasm of the egg. And the sperm is inserted inside the cytoplasm egg through a point. This point is called a focal point. So through the focal point, a single sperm is inserted inside the cytoplasm of the egg. So this technique is called intracytoplasmic sperm injection. Now this egg is converted into fertilized egg because the sperm is already entered inside the egg. Okay, so this fertilized egg is allowed to divide into 8-cell stage. Now this 8-cell stage plastomere is transferred to the transferred to the uterus for the development of fetus. Understand, dear students? So this method is called intracytoplasmic sperm injection. Understand? So these are some of the techniques or the methods involved in the assisted reproductive techniques. So intrauterine insemination, in vitro fertilization, zygote intrafallopian transfer, then intrauterine transfer, gamete intrafallopian transfer, and intracytoplasmic sperm injection. So these are some of the examples for assisted reproductive technique or technology. Okay. So now we are going to study about one important 3-mark question, surrogacy. Surrogacy meaning same. Some females or women agree or accept to carry the pregnancy for another one female. Okay, female who are ready to carry or accept to carry the pregnancy for another female is called surrogacy. Okay, so the baby is developing, so one woman's baby is developing in the uterus of another one woman. Understand of you? Understand? So this agreement is called surrogate. So just imagine this one person. Okay, this is another one female. So this female is ready to carry the pregnancy for this female. Okay, this woman. Understand of you? So a new baby is born for this woman. Okay, but she is the mother of this newborn baby. Okay, listen very carefully. So this person, woman A, here we are another one. So this A woman is ready to carry the pregnancy for this B woman. So a new baby is born, and the parent or the mother of this newly born baby is this B woman. Hope you understood. So this is called surrogacy. This is called surrogacy. Understand, my dear students? Then next we are going to study about one important 2-mark question: microtesticular sperm extraction. Microtesticular sperm extraction. This technique is used to prevent the male infertility. So one technique is used to prevent the male infertility. That technique is called microtesticular sperm extraction. Okay, say listen. Commonly, the person having low sperm or less sperm or without sperm, that condition is called azoospermia. Yeah, that condition is called azoospermia. So azoospermia meaning less count of sperm or less number of sperm or no sperm is called. The condition having very less number of sperm is called azoospermia. Understand? So here in this technique, a small incision. Okay, so a small incision or a small hole is made on the center region of the scrotum of male reproductive system. Okay, then through this hole, with the help of the microscope, the seminiferous tubules which are present inside the testis we can see. Okay, so we can see that the seminiferous tubules are present inside the testis. Understand you? Then the seminiferous tubule is dilated, and a small portion or a small amount of tissue is taken from the seminiferous tubule of the testis. Okay, so to produce this tissue is used to produce healthy sperm. So this technique is called intratesticular sperm extraction. So once again repeat. So in this technique, a small incision is made on the scrotum region, and with the help of the microscope, the seminiferous tubule which are present inside the testis is dilated. A small portion or small amount of tissues is taken from the seminiferous tubule, and with the help of that tissue we can produce more amount of sperm in the lab. Okay, so this technique is called intratesticular sperm extraction. Hope you are understood. Then, so now we have completed the first technique, first topic, assisted reproductive technology. Then, second topic is detection of fetal disorders during early pregnancy. Detection of fetal disorders. Fetal disorders meaning disorder affecting the fetus. Fetus meaning you all know. Okay, the baby or the embryo which are developing inside the uterus of the mother is called fetus. Sometimes the fetus may be affected by many disorders. So we can identify that disorders by using some techniques. So first we are going to study about one technique. Name of that technique is called ultrasound scanning. Ultrasound scanning. So this technique is used to identify the disorders in the fetus during the first trimesters. Already I told you that overall the total pregnancy duration is 9 months. These 9 months are divided into 3 trimesters. First trimester meaning first, second, third month. Then the second trimester meaning 4th, 5th, 6th month. Then 7th, 8th, 9th months are called third trimester. So this ultrasound scanning technique is used to detect the problems, okay, or abnormalities in the fetus during the first trimester. Okay, then actually when we compare this scanning technique with the other technique, it's a less risk technique, less risky technique. because a small pressure only given to the abdominal region and also in the vaginal region of the woman. Understand? So to take a scanned image, a small pressure is given to the abdominal region and also in the vaginal region of the pregnant woman. Okay, so no other risk in this technique. And when we compare this ultrasound scanning technique with other technique, here there is no radiations. They are not using any radiation in this ultrasound scanning technique. Okay, so 2 points I said about. First point, this technique is used to detect the problem during the first trimester. Second, I said a small pressure. Okay, it is a very low risk. So a small pressure only given to the where? To the abdominal region and also the vaginal region. And the third point I said there is no radiation. In this technique there is no usage of any radiations. Okay, understand? Then this technique, the advantage, this technique is used to identify the number of fetus inside the uterus. In some females uterus, sometimes 2 or more fetus can develop. There is a possibility is there. So to find out the number of fetus inside the uterus of the mother, this scanning technique is used. Understand, my dear students? Hope you understood this one. Then next technique is called amniocentesis. Okay, amniocentesis. Here amniocentesis technique means this technique also used to identify the abnormalities in the developing fetus. Okay, so in this technique, a small amount of fluid is taken from the amniotic fluid. Okay, amniotic fluid means the fluid present in the first extra embryonic membrane. Already we have studied four extra embryonic membrane. Okay. Amnion, chorion, allantois, yoxa. So these are the 4 extra embryonic membrane. Here the first extra embryonic membrane is called amnion. So inside the amnion is a double layer membrane. So inside this amnion, a fluid is present. This fluid is called amniotic fluid. So to do this amniocentesis technique, a small amount of sample fluid is taken from the amniotic fluid. Okay, so that fluid is used to do this amniocentesis technique. So if any abnormalities there, even the chromosomal abnormalities of the fetus also can be identified with the help of this amniocentesis technique. Okay, and this amniocentesis technique should be done between 15th to 20th week of gestation or pregnancy. Okay, between 15th and 20th week of gestation or the pregnancy only we can do this amniocentesis technique. Then next technique is called chorionic villus sampling. CVS. This is also the same technique used to find out or identify the abnormalities in the developing fetus. But here in this technique, here the amniocentesis technique, the sample fluid is taken from the amniotic fluid. But here in chorionic villus sampling, the sample is taken from the placental tissue. Placenta evolva, the tube connecting the fetus with the wall of the mother. Okay, wall of the uterus of the mother is called placenta. So in this, to do this chorionic villus sampling technique, tissues are taken from a small amount of sample tissue is taken from the placenta to detect the abnormalities in the fetus. Hope you understood this one. Then next, the last topic is fetoscope. Fetoscope meaning is a device used to hear the heartbeat sound of the developing fetus. Okay, when the fetus is developing inside the uterus of the mother, the heart is started to beat. Okay, so maximum 120 to 160 times the heart should beat in the fetus, in the body of the fetus. This is the normal heartbeat rate of the fetus. Okay, 120 to 160 times. Okay, per minute 120 to 160 times the heart should beat. So this is the normal heartbeat rate of the fetus. If any deviations are there, it indicates the fetus is not getting sufficient amount of oxygen. Okay, so if the amount of oxygen is not supplied properly to all the part or to the heart, it leads to some other problem to the fetus. So fetoscope meaning is a device used to hear the heartbeat sound of the developing fetus. Hope you understood this point. Then, so today we have completed many important 2-mark questions and 3-mark questions. For example, ultrasound scanning is a 2-mark question, separated 2-mark question. Define amniocentesis. Define chorionic villus sampling. So these are the 2 mark question and 1 mark question. Here in this amniocentesis, 1 mark question is there. In which time this amniocentesis techniques we have to do? 15 to 20 week of stage. Then here 1 2 mark question. What is the normal heartbeat rate of the developing fetus? 120 to 160 times per minute. So like that many 1 mark questions are there. Separately every techniques are 2 mark question. Here also every many 1 mark questions are there. All these assisted reproductive techniques are separately may be asked for 2 mark question. In define intrauterine insemination 2 mark question. Define in vitro fertilization 3 mark question. What is meant by XII? Okay, and like that many 2 mark and 3 mark questions are there. Many 1 mark questions also there. Understand, my dear friends? So hope you all enjoyed today's class, and today we have completed a very easy topic. Okay, both the topics are very easy. Okay, so all study and try to complete today's. We will meet in the next class. Thank you.